Autophagy plays a deceptively intricate role in the biology of cancer, not only within tumor cells themselves, but also within the surrounding stromal cells that make up the tumor microenvironment. This complex landscape, composed of fibroblasts, immune cells, blood vessels, and extracellular matrix, is not a passive backdrop, but an active participant in tumor progression, therapy resistance, and immune evasion. Within this ecosystem, autophagy can function in contradictory ways, sometimes suppressing cancer, other times enabling its growth. One of the most intriguing and troubling examples of this duality is the role of autophagy in stromal cells, which can inadvertently nourish and support malignant cells through a form of metabolic cooperation. In normal physiology, stromal cells like fibroblasts perform essential supportive roles, maintaining tissue structure and helping to regulate immune responses. When stressed by inflammation, hypoxia, or cancer-secreted factors, these cells may activate autophagy as a protective response. However, in the context of a developing tumor, this autophagic activation can be co-opted and redirected. Instead of clearing debris to maintain homeostasis, stromal cells under sustained stress begin to degrade their own intracellular components and release nutrients such as lactate, fatty acids, and amino acids into the local environment. Tumor cells, often under pressure from limited blood supply and high metabolic demands, eagerly absorb these resources, using them to fuel rapid growth and survive in otherwise lethal conditions. This phenomenon, sometimes referred to as the reverse Warburg effect, reveals a startling reversal of traditional tumor metabolism. Rather than cancer cells producing energy through glycolysis and dumping waste products into the stroma, the stromal cells themselves engage in high autophagic and glycolytic activity, effectively feeding the tumor. The cancer cells, in turn, operate more efficiently by taking up these metabolites and sparing their own resources. This metabolic symbiosis allows the tumor to expand while insulating itself from harsh environmental stress, including chemotherapy and immune attacks. The implication is deeply concerning. Autophagy, typically seen as a protective and restorative process, can under certain conditions fuel the very disease it is meant to suppress. Stromal autophagy doesn't just fail to stop cancer, it can become an accomplice. This reveals a major challenge in therapeutic targeting. Inhibiting autophagy broadly could theoretically disrupt this nutrient flow and weaken the tumor, but it also risks damaging healthy tissues and impairing immune function. On the other hand, stimulating autophagy indiscriminately to support anti-aging or cognitive health might inadvertently accelerate cancer progression in individuals with undiagnosed or early-stage disease. Moreover, autophagy's role in the tumor microenvironment isn't limited to fibroblasts. Tumor-associated macrophages and dendritic cells also rely on autophagy for their metabolic adaptation and function. In some contexts, this supports anti-tumor immunity by enhancing antigen presentation or enabling immune cell survival. In others, it promotes immune tolerance and helps the tumor evade detection. Again, the effect depends on the context, cell type, and disease stage. What benefits immune surveillance in one scenario might feed immunosuppression in another. This complexity issues a serious warning to those developing autophagy targeting therapies. Precision is not optional, it is essential. The success of such interventions will depend on the ability to selectively modulate autophagy in specific cell populations, at specific times, and within specific metabolic states. Tools such as nanoparticle delivery systems, tissue-specific gene editing, and autophagy flux imaging may help achieve this level of control in the future, but we are not there yet. Despite these challenges, the knowledge that autophagy contributes to nutrient cycling in the tumor microenvironment also provides a new therapeutic angle. Disrupting the metabolic cooperation between cancer cells and their stromal support could be a powerful way to starve tumors from the outside in. Already, some clinical trials are exploring combined strategies that inhibit autophagy in stromal cells while targeting metabolic vulnerabilities in cancer cells. Others are investigating how fasting or caloric restriction may temporarily disrupt this supportive interaction, sensitizing tumors to treatment. In conclusion, autophagy within the tumor microenvironment is a double-edged sword. While it can sustain immune function and prevent early tumorigenesis, it can also be hijacked to feed cancer cells, drive resistance, and deepen malignancy. This complex interplay underscores the need for a nuanced understanding of autophagy, not as a simple on-off switch for health or disease, but as a tightly regulated, context-dependent process. If we can learn to control it with precision, we may not only slow aging and neurodegeneration, but also cut off cancer's lifelines, one cellular pathway at a time.